Again, thank you for inviting me. Just a precision, I'm not anymore at University of Oslo. I am just associated with the University of Oslo.
I'm sorry.
I am at university. No, don't worry, don't worry. But I have not said anything before because the University of Oslo has hosted the project, and the project has been concluded in 2018, but we are still publishing results, therefore you are pretty okay when you say that I am affiliated to the University of Oslo. Anyway, I am in Italy at University of Ferrara, and I have just started today my lessons, therefore I cannot join you in person, sorry about it. Anyway, let's speak about the MEDPLAG project. This is an ERC supported project. And the goal of this project was to better understand the transmission modalities and ecology behind the plague. To do this, we have used several methods, not only one. That means archaeoanthropology and genomics. Therefore, we have analyzed ancient DNA, not from the victims, but from Yersinia pestis to better define the phylogeny of this bacterium. And then we have carried out other kind of study, climate, ecology, and modeling. And we have tried to interpret the outcomes of our results only on the background of the historical evidences. So let's consider that plague, you know for sure everyone, it's not a human disease, this is a zoonotic disease, and this disease is maintained all over the world in a plague reservoir. What is a plague reservoir? Is a place where the pathogen, Yersinia pestis, the host, mostly wild rodents, and the vectors, mostly their fleas, are present simultaneously. Human cannot be considered a reservoir because plague significantly increased the mortality rate beyond natural values. That means that the wild rodents in the reservoir has also some kind of resistance against plague. In the picture, in the map on the top, you can see in red the place where nowadays reservoirs are present. And you can see that they are present everywhere on each continent, in Asia, in Africa, in South, and in North America, in the United States, but not in Europe and in Australia. If you have a look to the other picture on the bottom of this slide, you can see the physical representation of the old world. And you can see that the reservoirs, which are in brown, are placed mostly or almost exclusively on the so-called desertic belt from the north of Africa through Asia. And you can also see that the ecological condition, and you can understand that also the climate condition of Europe is almost different from the desertic belt in Africa and Asia. So let's consider that, yes, I'm not surprised that at least nowadays there are no reservoirs of plague in Europe. But what happens in a reservoir, as you have understood, this is a desertic place. What happens when it rains a lot? This can happen. And in this case, it can be activated, a so-called trophic cascade. More rain means also more vegetation in the region, and more vegetation constitute more food for the wild rodents. Therefore, the population of the wild rodents expand, and they became bigger and bigger, and they can be generated even new population. And if they are infected with Yersinia pestis, that means that you have also more pathogens and more fleas which are able to transmit plague. But what happened if suddenly you have a drought or another shock, climatic shock that stops the presence of rain? You have less rain, less vegetation, but you had an enlargement of the population before. So many elements of this population can die. They have not enough food. Or they can try to get some food where there is more possibility, for instance, in the human settlements. And this is the point. They are looking, seeking for food, and they come in contact with very domestic or commensal rodents like the rats. And on this occasion, they can exchange fleas and pathogen, and they can infect the rats. The rats are very sensitive to plague, therefore this infection causes an heavy rat mortality. There are some description of this situation during the third pandemic, and observers are speaking about street littered, covered by cadavers of rats, black of rats. Therefore, it was not a phenomenon that could be overseen, at least in Asia. Well, what happened when you have this heavy mortality of rats? Their fleas cannot survive at a low temperature, therefore they switch to another host, and this other host can be also the human host. In such a way, you have a kind of epidemic among humans, which is sustained by the presence of the died rats. So this is a model, of course. It's not the truth, but it is a model. But this model fits very well with the third pandemic in Asia, in the USA, and in other places. Let's now have a look to what it's happened in Europe during the third pandemic. So in a study of 2019, we have considered the cases of plague in Europe starting from 1899. At least there was also some other cases before, but let's say from 1899. All these cases were notified to the CDC, therefore they are on disposal, and we have taken that from the repository and analyzed them, and we have placed them on the map. And what we have seen is that plague was mostly introduced with a steamship, and was therefore almost exclusively in ports. Don't be confused by the points. For instance, in Germany or in Poland, there are also ports. There are ports on major rivers. Therefore, we can really say that the majority of the cases and also of the outbreaks were not so heavy like in the medieval time, but we have, for instance, in Porto, about 100, more than 100 deaths for a plague in these periods. Therefore, there was also, we can say that there was also some outbreak. So, and since they were very aware about this mechanism, which was observed by Paul Louis Simon in 1894, that the rats were responsive for the transmission of plague, rats were deeply scrutinized everywhere in China, in America, but also in Europe. Therefore, we have also a lot of information. We have also considered some narratives, some medical reports from the period, and we have a lot of data also about the rats. Well, let's consider one example, and it is an example of Malta. Why Malta? Malta is an island in the south of Europe, very close to Africa and also to the desertic belt. Therefore, we can believe that it mimics like a bit the condition, environmental condition of the desertic belt. Even in Malta, they have analyzed more than 1,500 rats in 3 months. Therefore, there were rats, black rats, and also brown rats, and only 15 were infected. If you ask me, the reason is they were just came out from the ship. But of course, this is just a suggestion, just an idea. Anyway, Barnett, who was responsible of rat control during a plague outbreak, wrote that plague outbreaks always come to an end even if nothing is done to kill rats or their fleas. So, all considered, we can conclude that the role of the rats in sustaining epidemics was really modest, at least in Europe, at least in the period of the third pandemic. Considering these, we have taken into account also alternative ways of transmission. And we have, for instance, analyzed a case of plague transmission. If I remember well, there were about 30 cases in Glasgow, at the beginning of last century. So what is interesting, also in this case, there was no episodic recorded, and all cases could be reconducted to a previous human case so that we could reconstruct a sort of phylogenetic tree of the contagion. So this is an evidence that show how it is possible that a plague was really transmitted from human to human, and how for sure by contact, sharing object, or perhaps also some clothes. We have heard before that clothes, peste chiffonière, you have mentioned it, clothes can transmit plague, and this is also because they can contain ectoparasites. Of course, they can be the ectoparasites from the rats, but they can also be the ectoparasites of the humans. For instance, the body lice and Pules irritans. Pules irritans is not exclusive of the humans, but anyway, it is the major flea present in humans. And we have a lot of interesting reports about the living condition of the people at that time, but also about the presence in the schools of ectoparasites. They were all infested by ectoparasite in that period. It was not unusual, and if you consider in such quarters of Glasgow and of other cities in Europe, the presence of the ectoparasite, it's not something impossible. So, and another evidence in this direction. You have also said something, if I have correctly understood before, you have spoken about hygiene and cleanliness. Well, consider that the last cases of a plague in Europe were in 1945, at the end of the Second World War. And from the 50s, we had no more cases, just a couple of cases imported from outside, but no more really heavy situation. And this is a period which is also concomitant with the control of the insects. For instance, with insecticide, DDT was used massively in this time, but it was also the time of the introduction of domestic and private bathroom, and of domestic wash machine, the Hoover, and other methods. And this can be, of course, one of the causes of the disappearing from Europe of the plague. Of course, you have the same kind of civilization also in the USA and in China, but they have a reservoir, therefore it can happen from time to time. Some persons are infected, but in Europe there is no reservoir, therefore the chance of infection are really very, very reduced. And this was also and also the minimal number of cases in Europe in this period. It was also because of a very ideal sanitary policies of international prevention, due to many conferences held in Europe, in China, and everywhere, international conference, when they have decided the methods to avoid the entrance of plague in the different places. So this was very efficient, and this was also one of the reasons, perhaps, because plague stopped, disappeared from Europe. Well, what about the past? The project was mostly on the medieval plague, but we have to consider this mechanism in the actual, in the current situation, also to make some inference in the past. So we have tried to model the mechanism of transmission via human ectoparasites using mortality data. And we have compared this with the transmission by rats. And what we have seen is that the model of transmission via ectoparasites, human ectoparasites, is plausible. Of course, we cannot, this is just a model, we cannot be sure, but it is at least plausible, and therefore it is interesting to go also in this direction more in depth. Then we can also propose an alternative mechanism for the transmission and for the epidemics, which doesn't need the presence of local reservoir and of rats. Rats can be present at the beginning, but they are not necessary for the sustaining of plague. Consider that the previous model that we have seen can now be expanded to consider also the last circle with the transmission due to human ectoparasites or to contact from human, let's say, human chains of transmission. So if you consider this situation present, for instance, in a reservoir outside Europe, you need just to pick one wild rodents, one rats, or an infected people, or his clothes, or his personal objects, and put them on a ship and send away somewhere, and then plague can recirculate by means of human chain of transmission and even mimic the presence of a reservoir in another place. So this is again another model. But we have tried to found some support for this model. For instance, we have analyzed the presence of some climatic change in Europe during the period of the second pandemic and in Asia. We have found no signal in Europe that can justify the presence of a local reservoir, but we have found a lot of signal in Asia, which precede epidemics in Europe of about 15 years. If you consider that in Constantinople, which can be considered, together with Venice, the door from the Orient to the Western world, in Europe, during the first pandemic, plague struck on average every 15.4 years. This is interesting, and of course, it's just a suggestion, but it can be a good suggestion in our direction. So, and now let's consider the phylogeny of Yersinia pestis. Here I can present you the first study that we have carried out in 2010, and it was the first phylogeny with some strains of ancient DNA obtained from victims of the second plague pandemics. And in particular, the strains were those of Bergen-op-Zoom, Ber, you can see it in green, and Erfurt and Saint-Laurent de la Cabrillerie. Okay, Yersinia pseudotuberculosis is the antecessor of Yersinia pestis. From Yersinia pseudotuberculosis to this point is the branch called the branch 0. And then you have branch 1, who has originated the third pandemic at the end of the branch. And on the other side, branch 2. So already in this study, we could see that the strain of Bergen-op-Zoom was placed on branch 1, and the other one were quite different. We could not well identify their position, but we tentatively placed them on this point, which is called the Big Bang, which gave rise to the polytomy, that means more branches. Okay, from 2010 until now, there have been published many papers, not only from our groups, but from the group of Krause and from other groups. And we have made a summary, and so I can propose you this, which is the most updated phylogeny of the second plague pandemic. What it is clear now is Saint-Laurent de la Cabrillerie can be placed a bit more closer to Bergen-op-Zoom, together with all the other strains of the Black Death. So, this is also another important point. From the Black Death period, we have the appearance of another branch. Let's call it the post-Black Death, the post-BD branch, to better understand. So it is clear that this branch was settled in a new reservoir, but the question is, where is this new reservoir which hosted all the population of the second pandemic, or almost all, not all? Well, in this paper, just published a couple of weeks ago, we have proposed to analyze the phylogeny, keeping in mind two hypotheses. The first hypothesis is that plague was introduced into Europe just once at the time of the Black Death, and it developed in Europe in a local reservoir. It was proposed by Anna Carmichael, but it was an hypothesis just published some years before, that a local reservoir could have been on the Alps, and to be more correct, on the French side of the Alps. There are also other proposals, other suggestions, for instance, that the local reservoir was in Germany. Then, after this idea, plague was later reintroduced to China to give rise to the third pandemic. But let's concentrate now on the local reservoir. This is the first hypothesis, the one introduction model. The second hypothesis is the multiple introduction model. This is more supported by our group, but not only. For instance, Morozova et al. Have published a paper recently, also in support of this hypothesis. But anyway, it is an hypothesis. Just consider it as it is. So, based on what we have said before, we consider that plague could have been hosted in a reservoir in Asia and transported from time to time, exactly as it was at the time of the third pandemic, to Europe, and then it could have been recirculated by means of human chains of transmission from a place to the next due to trade, travel, exchange of goods, clothes, and so on. Let's now consider the single part of this branch. So you can see that Krause and his groups has published the most ancient genome of the period, which is a strain from Russia on the Volga, exactly, to be exact. And it precedes the Black Death. Then you can see that there were also other strains, Bergen-op-Zoom, we have spoken about it, which is in the Netherlands, London, and Bolgar. Bolgar is again on the Volga. So to explain this situation, we have proposed that plague was introduced on the food trade routes. Why this? Well, we have asked ourselves what is the land of darkness, and by doing some research, we have found that it was the name used by the merchants of this period to define the place where the best fur came from, and in general, the trade routes of the fur. And we have seen that there were two principal routes from the south and from the north, which can justify the presence of the plague during the 14th centuries, not only during the Black Death, but also in a later period. And that means that these strains are more close to the place where the main reservoir of branch 1 was placed. But what about the post-Black Death strain? So after the model of the local reservoir, you have to consider that it remains in Europe. And this is the reason because we have painted it in blue to distinguish the other hypotheses which consider their introduction from the east, which is therefore painted in brown, just to distinguish them. So the hypothesis of the persistence of plague in a reservoir, in a local West European reservoir, is also based on the presence of several strains or genomes found in Middle Europe. There is some uncertainty about the datation of the sample, and for instance, you can see that the strain of Cambridge can be dated to the 15th until the 17th century. Therefore, it's not so easy to give an explanation. But for some strains, like the strain which are represented on the right, I don't know if you can see my arrow, but they're present on the right, and they're marked with the 30 years words clade. We know very well that they were of the period of the 30-year war, and in some cases there are also some soldiers of this period. And you know that it was a great period of trade, travel of troops or soldiers everywhere, and they take with them not only plague but also other epidemics. During this period, the plague was also imported to Italy, and we know in Italy very well, we say that there were the Lanzichenecchi who take the plague to Milan. So therefore we can explain this part of the phylogeny also with this historical data. As for the end of the second pandemic, we have, of course, much more information, also historical information. And for instance, the plague of Marseille is well known to have been imported from a ship, the Saint Antoine, who at least was in Syria. We don't know if it is the region. We know that the strain that we have analyzed from Pest originated from the Anatolian part of the Mediterranean basin. And then you can see that also the most recent sample that we have came from the Caucasus. But we have also, thanks to the work of Morozova et al., some strain coming from Rostov, which is closer to the Black Sea. And they are very interesting because they are, you can see on the bottom of the picture, they are from the same cemetery, but they show two different genomes. And this is also possibly a signal of being more closely to the reservoir, to the actual reservoir. Therefore, at least for this last part of the phylogeny, I think that there is not many doubt that it was imported from the East. And if it was, the reservoir was in the East, probably it has remained in the East from the time of the Black Death. I cannot say you where, in the Middle East, in the Central Asia, somewhere, I don't know. Well, in the same paper, we have also analyzed another kind of evidence. We have actualized the data of other scholars, and we have seen that at the end of the two main plague pandemics, that means the first and the second, there were two convergent mutations in the genome of Yersinia pestis. A deletion of 49 kilobases, a large part of the chromosome, and the depletion of the plague gene. Depletion means that since this gene is on a plasmid, that they had less plasmids containing this gene. And actually, these two convergent mutations may represent a loss of virulence of the strain. Therefore, Sousa et al. Has interpreted this signal as the possibly reason for the end of the two plague pandemic. There is actually another explanation, which is an evolutionary explanation, and which is based on the observation of the modern strain. Plague tends to reduce its virulence to have a fitness advantage. This can happen when it adapts to a new host, for instance, or if the host die too much. So they try to increase at least the time of death so that, the bacterium has more chance to be transmitted to someone else, to find another host. And so we proposed very, very tentatively that this mechanism was at least a sort of evolution which has developed during the pandemic when the bacterium was mostly present in humans, and therefore that humans are the new host. And of course, we have only this data, this genetic data in support of this hypothesis up to now, but new genomes from Larrier in France, from the French Alps, do not show this plague depletion, and therefore it is difficult to consider that it was an adaptation to a new host in this putative West reservoir. So concluding, Yersinia pestis phylogeny should always be interpreted in the light of historical context and of possibly of other evidences. And I suggested that alternative theories and plague transmission are plausible and they should be investigated more in depth. And to consider them would perhaps also avoid future epidemics and pandemics. Thank you for your attention. And this is the group of MEDPLAG.